Hello and welcome back to Windows Seat Studies. Today we are going to learn about oxidative bisulfite sequencing. So in the last class we learned about bisulfite sequencing and today we'll be learning about oxidative bisulfite sequencing. So from the name it's clear that oxidative bisulfite sequencing is an improvised version of bisulfite sequencing. But why are we improvising the bisulfite sequencing? It means that there must be some drawbacks, some limitation of bisulfite sequencing which we need to overcome, and that is why we invented a new sequencing method, and that is oxidative bisulfite sequencing to detect the DNA methylation. So what is the drawback? Let's learn about it. So for a quick recap, in bisulfite sequencing, it is able to differentiate between cytosine and 5-methyl cytosine. Okay, so what happens that in the bisulfite sequencing, cytosine is deaminated and it is converted into uracil due to sodium bisulfite. Okay, but sodium bisulfite cannot work on 5-methyl cytosine, and that is why 5-methyl cytosine is read as cytosine in the bisulfite sequencing. So the drawback of bisulfite sequencing was it can differentiate between cytosine and 5-methyl cytosine, but it can't differentiate between 5-methyl cytosine and 5-hydroxymethyl cytosine. Why? Because sodium bisulfite can only work on cytosine and it cannot work on 5-methyl cytosine or 5-hydroxymethyl cytosine. But there's a there's a thing that you need to know here, that sodium bisulfite can only work on cytosine and 5-formyl cytosine. Okay, it can only deaminate both of this. So it can't work on 5-methyl cytosine and 5-hydroxymethyl cytosine, and that is why, just like the 5-methyl cytosine, 5-hydroxymethyl cytosine is also read as cytosine in the bisulfite sequencing. So now the glitch comes here, that bisulfite sequencing, the drawback is that bisulfite sequencing can differentiate between unmethylated and methylated DNA, but here comes the but it cannot decipher or differentiate demethylated DNA. Okay, by the meaning of demethylated DNA, it means that it cannot recognize 5-hydroxymethyl cytosine. Okay, so for overcoming this drawback, for overcoming this limitation, we introduce oxidative bisulfite sequencing. So it is clear from the name that there is one step involved in the bisulfite sequencing, and that is oxidation. So all of the steps are same, but we are going to oxidize 5-hydroxymethyl cytosine and convert it into 5-formyl cytosine by potassium perruthenate. So the reagent we are going to use is potassium perruthenate. Okay, this double R in the perruthenate. So we are going to use potassium perruthenate, and this potassium perruthenate is able to oxidize 5-hydroxymethyl cytosine to 5-formyl cytosine. And once this is converted, on this, the sodium bisulfite can work and it converts this 5-formyl cytosine to uracil. Okay, and this uracil is read as thymine in the sequencing. In the sequencing steps, it is read as thymine. So what happens in oxidative bisulfite sequencing is that in oxidative bisulfite sequencing versus bisulfite sequencing. So in bisulfite sequencing, the cytosine is read as, so cytosine gets converted into uracil, and that is why it is read as thymine. Okay, so let me write it as follows. So in the bisulfite sequencing, so this is the original nucleotide, and this is converted nucleotide, and this is what is read in the sequencing. Okay, so cytosine is converted into uracil and it is read as thymine, while 5-methyl cytosine is not converted into anything, and that is why it is read as cytosine only. While 5-hydroxymethyl cytosine again on this sodium bisulfite can't work and it is read as cytosine. And 5-formyl cytosine, it can be converted into uracil and it is read as thymine. But let's not focus on 5-formyl cytosine here because we are just comparing cytosine, 5-methyl cytosine and 5-hydroxymethyl cytosine. Means the unmethylated DNA, okay, the methylated DNA and the demethylated DNA. Okay, that is what we are comparing. So this is the case in the bisulfite sequencing. So what happens in oxidative bisulfite sequencing? The cytosine is converted into uracil and that is read as thymine. So cytosine is converted into uracil with the help of sodium bisulfite, obviously. Well, 5-methyl cytosine is not converted into anything because sodium bisulfite can't work and it is read as cytosine only. While 5-hydroxymethyl cytosine is converted into 5-formyl cytosine due to the oxidation step, and on this the sodium bisulfite can work and it converts it into uracil, deaminates it, and it is read as thymine. Okay, so it's one more step that it is converted into uracil. So this is a step and then it is read as thymine. Okay, so this is the difference. So now you can differentiate between 5-methyl cytosine as well as 5-hydroxymethyl cytosine in the oxidative bisulfite sequencing, and that is why oxidative bisulfite sequencing, this is how oxidative bisulfite sequencing is able to overcome the limitation. In the next video lecture, we'll learn about we'll solve a problem and we'll also learn about tab sequencing. Okay, till then, see you. Bye bye.